spelled R-Y-A-N, last name H-E-M-M-E.
Ryan, and your position, what's it called?
I'm an entomologist with the Centers for Disease Control at the dengue branch in Puerto Rico, San Juan, Puerto Rico.
Okay, and so what are you doing out here today?
Well, today we're actually out, and one of our main objectives when we arrived in Hawaii was to evaluate two different traps that can be used for mosquito surveillance. So today we're looking to see how the autocidal gravid ovitrap as well as the BG trap can be used for surveillance and monitoring of Aedes albopictus in Hawaii.
How many traps do you have
Okay.
put around, and what are you doing with them?
So in this area that we're working in right now, we have a total of 20 BG traps and 20 Ago traps. And these traps will be out for the next couple weeks, and to get a good idea of the actual mosquito composition and density of mosquitoes in this area. And ideally what we'd like to do is repeat this study at other locations where there could potentially be dengue transmission to see how the sites are similar in density and abundance, and then how they are also different.
Okay. And did you explain the difference between the two traps? What's a BG trap?
Sure. So we're using the BG trap and the Ago trap. The BG trap is actually collecting mosquitoes that are looking for a host to bite, and they're attracted to the trap visually with the black sock around it. That's how I refer to it. So they're actually looking for a host or potentially some place to hide, and then they get sucked into the trap with a fan that's powered by a battery. The Ago trap is also visually attractive to the mosquitoes because it is black, but in addition to that, it has a water hay infusion in it that the mosquitoes can actually smell. And so the mosquitoes are smelling that, and they're actually flying to that trap in hopes to lay their eggs. So they fly into that trap, and then they'll get stuck on a sticky membrane that's on the inside. And then once we collect the mosquitoes, we can take the samples back and actually do some testing on them for if they are infected with the virus or not. So it's very critical that any integrated vector management program, they have the operational and vector surveillance research that can really inform the epidemiologists and entomologists about vector management practices as well as possible ways to mitigate the outbreak. So what we're doing is trying to add to that base of knowledge and provide that research that can help inform the public health officials in the best decisions to make.
Okay. Is there anything else I didn't ask you about what you're up to today that you'd like to comment on?
No, but I would like to say what an honor it has been to be invited out to Hawaii and work with Hawaii Department of Health, and especially Dr. Hasty. It's been a wonderful collaboration the last couple days, and I'm really looking forward to the work we can do over the next week.
And how long are you guys going to be here?
Currently we're scheduled to be here for 2 weeks, but that could also change. It really depends on what the CDC thinks and what the Hawaii Department of Health wants to do. We are actually here to help Hawaii in any way possible. So that is our goal.
Okay. And just quickly, what kind of mosquitoes are you finding?
So the majority of mosquitoes we're finding here is the Aedes albopictus. This is the dengue vector in the area. We also find another Wyomia species, as well as I think we found Aedes japonicus and some Culex species. So we're really interested, of course, in Aedes albopictus. Then if we do find Aedes aegypti, which we have not found in this location, but we have found in other locations, those are the two most important mosquitoes that we're actually interested in finding out where they are and the density of those mosquitoes, the abundance of them. Yeah, it's been really nice working here in Hawaii. Everyone that we've visited has been very cooperative with our research, and they've actually been really interested in, the mosquitoes that transmit the virus, learning about their biology and the current state of the outbreak.
Now, earlier today, we were in your little makeshift lab over at
Health Department.
The health department. Thank you. And what was the tech doing there?
Gilberto Felix is our biologist, and he was sorting mosquitoes, identifying Aedes albopictus and any other mosquitoes that we find from this site. And then he'll sort them by species as well as sex, and then we put them in a vial And the ones that we're interested in testing, so primarily Aedes aegypti and Aedes albopictus, will be sent to the CDC dengue branch for virus testing.